What is the hippocampus? Where is it, and why has it historically been considered central to memory?
The hippocampus is you'll often see it described as a C-shaped structure in the brain. So in the human brain, it's kind of deep at the base of our brain, underneath the striatum and thalamus and all of our cortex. In the rodents, it's kind of in a similar place. And it has been considered central to memory from 1950s from a study in a classic patient named Henry Molaison, and he goes by HM in all of your textbooks. So what they found with HM was that he had intractable epilepsy, meaning that his epilepsy could not be cured with medication. And so the only solution was to resect or take out his hippocampus, which was the source of the epilepsy, and they took out some of the surrounding tissue. And no one at the time knew what would result from this. His epilepsy did stop, but Brenda Milner, who was a neuroscientist who worked with the neurosurgeon who performed the surgery, William Schofield, started to follow up with HM and visit his house and see how he was doing. And they found that he could no longer form any new memories. So every time, well, let me clarify, any new episodic memories. So anytime that they would come to his house, he would be like, I've never seen you before. Who are you? Why are you here? But he could still pick up tasks that they taught him. So they taught him this backwards star drawing in a mirror. And every time they came to visit him, he got better and better at that task, but he could never remember having learned it. And so this was kind of the first clue to the whole world that the hippocampus is really important for forming new episodic memories. And an episodic memory is one that has events and details and people, and you can recall it and tell it like a story, as opposed to a procedural memory, which is muscle memory, which is learning how to do something with your body, which you don't necessarily need to be able to tell a story to do, you just learn how to do it. And so the hippocampus really underlies this episodic memory component. And yeah, I think we can talk a little bit more later about kind of where we are today in terms of what we think it does. That's a little bit more complex than this.
So episodic memory and procedural memory, are these sort of the two types of memory, or they're sort of different other subsets of memory, if you know?
Yeah, so the kind of in the psychology world, the two classic types of memory are declarative memory and procedural memory. You also have semantic memory, which is when you learn facts. Like you might learn them through initial episodes. Say you take a class as a child, you might be able to recall where you were sitting in the classroom, who you were sitting with, what the teacher looked like, what the room looked like, any feelings that you were having that day. But you also learn some information during that experience that then can be stored and remembered kind of independently of that visual scene that you're remembering from the experience. So that would be semantic memory, that knowledge that you recall. And the hippocampus is important. So declarative memory, the subsets are episodic and semantic, and hippocampus is really important for the initial learning of the semantic information and the memory formation and recall of the episodic memory. But after a time, that semantic information gets transferred somewhere else in the brain. The classic theory is that it goes to the cortex, and then you can recall that independently of the hippocampus later on.
So I presume, like any brain structure, the hippocampus is well connected to other brain structures. Are there any specific brain structures that it's more interconnected with, or anatomically and neuronally?
It is extremely well connected with almost the entire brain. So there's a really beautiful paper showing this from the kind of early 20s. And to back up and give you some context for what I'm about to say, one of my favorite phenomena in the hippocampus is the phenomena of replay. So replay is this process where your hippocampus literally recalls the specific order of events in an experience at a very time-compressed scale, and can then send that information to the whole brain to pull out the visual information and the smells and the sounds and all the things that were initially part of the experience. And so the study in the early 2010s used monkeys, and they recorded these replay events in the hippocampus, and then they did fMRI of the monkey's brain. And what they saw is that the entire brain, except for the primary sensory cortices, lit up at the time of these ripples, these replay events. And there's this kind of EEG phenomenon in the electrical activity of the network that's called a ripple, and we can kind of use those as a biomarker for these events. Now, the reason that the primary sensory cortices did not light up is because if you think about when you are recalling an experience, you sort of become introspective, right? You tune into like your mind's eye as opposed to paying attention to the external world, even if it's only for a moment. And so the brain systems that pay attention to the external world and are kind of encoding new experience have to become quiet while you enter this introspective state of remembering something. So I think the thing that that study highlights is also how incredibly interconnected the hippocampus is with the rest of the brain that, it can go boop and reactivate this memory and basically the whole brain lights up. But it does have, specific connections to certain areas that we can talk about if you're interested.
So, little quiz for me. The hippocampus plays a role in semantic memory. Is that correct, or have I butchered that?
Primarily episodic memory. Yeah.
Episodic memory. And so I presume those are the types of memories that disappears when the hippocampus is damaged, right?
Yes, especially the formation of them. So that's why HM had what we call anterograde amnesia, where he couldn't form new memories, but he could remember things from his childhood when he was 8 years old, let's say. So these super long-term memories seem to get somewhat stored somewhere else, although I think it's still a little bit unclear. So kind of more contemporary research suggests that you still need your hippocampus even to remember the details of those experiences a really long time ago, even if you're able to say, let's use the classroom example again. You might have the semantic information stored. I went to school at, I don't know, Williams High School, right? You might have that semantic information, but if someone said, can you describe to me your first or your favorite third grade class at this, in this school district, you would not be able to do that if your hippocampus was damaged. So you can still have that kind of factual memory, but not the vivid recalling of a specific sequence of events.
So is that also seen in patients with dementia, or is that too much of a stretch or generalization?
No, it's not too much of a stress. So one of the first symptoms of dementia is an impairment in spatial navigation. And I can speak to this from personal experience that this happened with my mother-in-law, that the kind of the first sign that something was wrong is that she drove half an hour in the wrong direction somewhere and had no idea where she was. The reason that this is because the hippocampus and the cortex that it's most strongly connected to, the entorhinal cortex, where it receives its kind of filter of a lot of sensory inputs and navigation inputs, those degrade first. They show the first marks of neurodegeneration. And so in dementia patients, what you often see is that they become impaired at navigating, at remembering where they are and how to get to certain places. This is a fundamental function of the hippocampus. They also have trouble forming new memories. And so, again, from firsthand experience, what I saw is that, she would tell a story and then 5 minutes later tell the same story and have no recollection of having just told it. So that kind of online formation of those new memories to say, we just talked about that and I can remember having just talked about that, so I'm not going to do it again, that seems to fall apart. But dementia is very complicated. There's a lot of different types. There's a lot of individual variability in what parts of the brain it affects. Sometimes you see the plaques associated with Alzheimer's disease. Sometimes you don't, and you still see the symptoms. So I think it's still, unfortunately, still really unclear how to target it and interfere because we still don't really understand what is exactly going wrong.
Before we get on to place cells and the HPC EC, I have a sort of out of the box question. So semantic memory can be sort of, let's say, linked to facts, let's say.
Mm-hmm. Mm-hmm.
Would you say episodic memories, then there's sort of an emotional influence on episodic memory, if that makes sense? Because semantic is, like I said, facts, it is what it is. But with episodic memory, you can take into account the context of the environment you're in,
Mm-hmm.
the sounds, the smell, the music, whatever it is, to sort of influence and have an effect on that memory. Is that sort of a fair summary or a stretch again?
No, I think that is a fair summary. Episodic memories often do have an emotional component. And one thing that I've been fascinated by, and is kind of what I study, is that if you think of any of your strongest memories in life, they're always the ones that were either the most positive or the most negative, right? They're always the ones that have the strongest emotional component, your college graduation or your wedding, super rewarding, super positive, rich memories, or maybe a car accident or something, the loss of a loved one, really kind of traumatic, sad memories. Those will be the strongest. And this is a documented thing both in animals and in people, but we're still figuring out the reasons for why that is.
I mean, do you think this is a single variable, but do you think it can be linked to, first of all, dopamine when it comes to sort of happy memories and sort of those sort of emotional, powerful memories? And then maybe it's the hippocampus linked to, let's say, the amygdala with regards to fear and emotions. That, I mean, it comes back to your initial points about how interconnected the hippocampus is, but have people sort of interrogated those circuitries from a sort of a clinical experimental perspective?
I know less about the clinical experimental perspective, although I believe there is a group that uses kind of deep brain stimulation in the amygdala to try to disrupt traumatic memories and PTSD,
Mm,
and they've seen some success. So there is this kind of idea that the amygdalas may be overactive. And I actually recently learned that people with PTSD have more myelin, more of the kind of fatty sheath that insulates axons and improves connectivity. So one possibility is that in folks with PTSD, they're like the hippocampus and their amygdala or whatever brain region is important for this strong, fearful state is too well connected. There's too much connectivity, there's too much activity. And so, they're not able to kind of turn down that fearful response when they see cues or sounds that remind them of that initial traumatic experience. So, if in response to dopamine, yes, there's been a ton of work on dopamine. And I want to clarify for the listeners that dopamine is not just the happiness juice in our brain. It is a very complex signal that is responsible for a lot of things. So the kind of classic knowledge in the field that we know it definitely does is this thing called reward prediction error. So this is the difference between what we were expecting to get and what we actually get. So let me give you an example of this. Let's say my doorbell rings and I'm expecting a pizza delivery. That doorbell is a cue that predicts the pizza delivery. So I get really excited. I would, in my ventral tegmental area, which is one of the homes of dopamine neurons in the brain, I would have a spike in activity at that cue. If I get the pizza pie that I'm delivered is exactly what I was expecting. It's, hot and cheesy and has all the pieces and it's the right size and it has the right amount of pepperoni. I would actually have no dopamine firing in my brain. And that is confusing if you think of dopamine as just the happiness juice. I do feel happy. I do feel excited that that pizza is there. But because I'm expecting it, dopamine neurons, now, of course, there's some variability, there's heterogeneity and all these things, they actually would fire none because there's no prediction error. The prediction is exactly what I got. Now, let's say they deliver 2 pizzas instead of 1, then I would get dopamine firing because I would have a positive prediction error. I would get more than I was expecting. Or if they come and deliver and I only got half a pizza, I would get a dip in dopamine firing because that would be a negative prediction error. It was worse than what I was expecting. This is a little bit of an oversimplification. There's some evidence that there's a difference between how dopamine neurons are firing and how dopamine is actually released in downstream structures. So we know that dopamine also encodes the value of actions, it encodes motivation, it helps promote movement vigor. You're going to run faster towards something you want if you have more dopamine versus less. So it's very complicated. But there is some evidence that in the hippocampus, when you have dopamine, you have stronger encoding of rewarding events. And if you disrupt dopamine signaling, you can kind of reduce that encoding. But the hippocampus actually doesn't receive that strong of dopamine innervation from the VTA, this area that I mentioned that has the dopamine neurons. There's another brain area called the locus ceruleus that actually co-releases both norepinephrine and dopamine. And norepinephrine is thought to be this sort of like saliency signal. Something is new and interesting. Even if it's not rewarding, it's something to pay attention to. You get norepinephrine. And so maybe this area is kind of controlling the dopamine in hippocampus. There's some evidence for that, but it's still a little bit of a mystery. And I think that you can represent reward in the brain and positive memories without it being dependent on dopamine. So it's a little bit more complicated than that.
Yeah, I think especially in the modern world, dopamine gets oversimplified, as you said, as that sort of the happy molecule.
Right.
It's also related to pain and pleasure. That's the sort of quote that Anna Lemke here at Stanford sort of wrote about. Um, yeah, before I sort of deviate on dopamine and everything about that. When your review talked about place cells,
Hmm.
and place cells, are they located in the hippocampus?
They
And
are.
what are they?
Yeah. So place cells were discovered in the 1970s by John O'Keefe, who was just putting electrodes in the rat hippocampus and having the rats do different things. And what he saw is that the cells would most reliably fire whenever the rat visited the same place in a box. And so they are now very famously known as place cells because they will fire in one or a few specific locations in space. So in my home office here, if I were to always go to the northwest corner, certain cells in my hippocampus would fire. If I went to the southwest corner, a different group of cells would fire. When I'm sitting at my desk, a different group of cells would fire. So they encode where you are in space. That is an undeniable fact. What is more debated is whether they encode more than that. And I think it depends on who you ask. Some people are really still kind of hard and fast about this spatial purism, but most of the field agrees that they do a lot more than that. And so the way I was trained to think about place cells is that they encode events in our experience, and if you string together these events, you get an episodic memory. And so they are sort of the scaffold or the backbone that you can link other parts of experience to and then put them in order to get a memory. And so kind of getting back to your question about what is the difference between episodic and semantic memory? Is it emotion? It's partly emotion, and place cells will also, encode emotional events differently than non-emotional events. But I think even more fundamental than that, it's this idea that my PhD advisor calls generativity. So this is the idea that the hippocampus can also underlie imagination. So there's a fundamental similarity between imagination and episodic memory in that you have to go into your mental imagery, into your mind's eye, and simulate a future experience when you're imagining something, right? Similarly, when you're recalling an episodic memory, you go into your mind's eye and you simulate, or you kind of relive that previous experience and can imagine it as it was. And there's modern evidence that humans with hippocampal damage, in addition to being impaired at forming new memories, they also can't imagine things with the same richness that healthy people can. And so if you ask somebody with hippocampal damage, imagine a vacation on the beach. And you can ask them this if they've never gone to the beach, right? But they've seen the beach in movies, they've read about it, they've seen pictures. A person with hippocampal damage, let me say a person without hippocampal damage will say something like, I'm sitting on a beach in Jamaica and the water is beautifully blue and the sky is clear and I'm reading a book and someone walks by and offers me a drink. And I buy the drink and it's a banana daiquiri or something. And so they're describing a sequence of events in order and the richness of the experience. They're describing the imagery. They're describing how they feel. They might say, I feel so relaxed. They're putting this emotional component to it. If you ask someone with hippocampal damage, describe a vacation on the beach, they'll say something like, I go to the beach, it's blue, it's hot, and then I go to dinner. So there's no richness, there's no details and sequence of events. They're just saying facts, right? They're saying, I go to the beach, it's blue, it's hot. These are facts that you could say about the beach that have nothing to do with an individual episodic experience. And so it's that ability to link together those little pieces that is what the hippocampus is fundamentally doing. And so that's where place cells come in. They are those individual pieces, and the hippocampus links them together in sequence. And we see this kind of at a variety of timescales. So maybe I'll pause because that was a lot of talking, and get I don't want to go past two fundamental questions.
I have 3 or 4 questions that I've wrote down.
Yeah, go ahead.
Would you say that place cells are like, in the simplistic of terms, coordinates essentially of locations? So they represent sort of coordinates of locations. Is that too simplistic, or?
I would say that's not correct because let's take the example of I'm in my home office here. If I were to repaint the walls and move the furniture, the place cells would fire differently. So that means they are not coordinates because they're not just reliably firing in the same geographical place in space. They're firing in a contextually dependent way. So if the context has changed, the place cells will change how they're firing and a new group of cells might become active. So the other thing I want to clarify is that in any given experience, something like 30 to 50% of neurons, again, this kind of depends on the conditions of the experiment, will become active as place cells. But there's all these other neurons that are not firing as place cells. And that subset of neurons that's active as place cells will change experience to experience, so that every time you change the experience, you change the context, it's like a hand is coming down from space and plucking up a different group of cells to become active.
Okay, so then that leads to my second point, which is a question, which is going to sound really stupid, but
never.
If you've been to, let's say, multiple different countries in the space of 2 years, and you've had all of these different events, but you only have a finite number of place cells, right? So are we talking about a different combination of place cells being activated? What's going? I'm thinking this from a very sort of quantifiable perspective. That's why.
No, this is a really important question. Right. If you had to have a different set of place cells active for each of those experiences, you would run out of storage space in your brain pretty quickly,
Hmm.
right?
Hmm.
That is not what happens. The same place cell can have different activity in a completely different location in space. It can have slightly different activity and different experiences. So you can reuse the same group of cells over and over and over and over again.
Mm.
And it's the combination of different firing patterns across the whole population that distinguishes the different experiences. So let's say you
Mm.
have, place cell A and it fires in my office, and then I go to the beach. That same cell could fire on the beach, but
Right.
it
So
will, yeah, good.
sorry, just on that, because
Yeah.
this is going to sound really weird, but like, do you think there are a cell, like certain subsets of place cells, which are then related to certain contexts? So let's say place cell A is always going to be activated on a beach, but it's a different beach depending on the location of that beach and the context of that beach. And then maybe there's going to be a certain subsets of place cells which are activated when you're in an academic environment, but that academic environment might be different. Like I'm now in Stanford, but maybe when I was in Cambridge doing my PhD, the same similar sort of place cells. Like obviously these aren't sentient cells and sentient beings, but it could be influenced by the context in similarities,
Yes.
let's say.
Yes, I think that is true. I think if there is contextual similarity, you are more likely to get a similar set of cells active. So yes, I think that is absolutely a thing because let's say that cell that's firing on the beach, it is, kind of being triggered by a set of inputs that signal the context of beach. It might fire slightly differently on one beach versus another, but there will be some similarity there. So the way I think about it is that the specific experience and the way that, say, one beach is differentiated from another, or the way Stanford's differentiated from Cambridge, is the specific pattern of spiking between cells at the whole population level. It's not that the same cell even needs to fire every time you go to that beach, because there's this other phenomenon in the hippocampus that, let me repeat that sentence. That was a little. There's another phenomenon in the hippocampus that cells will do what we call drift. So that means that let's say you're on vacation and every day of vacation you go out to the same place on the beach where the beach chair is and you sit there, you might have a set of cells that fire for that experience, but they will fire slightly differently every single day. So every repeat experience, you could be sitting in the exact same place on the beach, you could be doing the exact same thing, and they will have slightly different firing patterns. Now, that is actually kind of useful, right? Because if we think about episodic memory again, your brain needs a way to differentiate the different days of the vacation. If your cells fire the exact same way every single day of the vacation, how would you know that it was a different experience, right? So this might actually be beneficial, and one of the theories behind this is that this kind of drift or slight change in activity from day to day is encoding the time or the passage of experience. There's actually evidence that it's passage of experience more so than clock time. But that also means that you can't think of the firing of place cells as coordinates or as only encoding location, because same location, they don't fire the same way.
So that's so essentially with place cells, text matters. Is that
Yes, very
okay?
much so. Yep.
Okay. So what about grid cells and the hippocampus and its role with I'm going to butcher this pronunciation. I'm not even going to say it. You say it. The hippocampus and the EC.
Entorhinal cortex.
There we go.
Yes.
I'm just going to call it the HPEC or whatever. But yeah, please talk to us about that.
Right. So the entorhinal cortex is both the primary input and output of the hippocampus. It's in a loop. So you'll often hear about it as the input because, I don't know, that's how it was traditionally thought about, but the hippocampus also projects back to the entorhinal cortex. So they're in a loop. In the entorhinal cortex, there are about 15% of cells that are called grid cells. And they get a lot of attention because their firing pattern is so mathematically beautiful. It can be described by mathematical models. It kind of fits this principle of maximally dense packing. So if you think about marbles in a jar, they'll all kind of slot together so that they're in little triangles, right? If you've got two circles next to each other, the only place that a marble can sit is right between them, and it makes a triangle. You put a whole bunch of triangles together, you get a hexagon. And so grid cells fire in this exact pattern. They'll fire in a hexagonal grid, which means that if you think about a rat running around a square box, there will be little pockets of activity or little bumps of activity in a hexagonal pattern across that box. The edges of the hexagon will not be perfectly aligned to the edges of the box. They'll actually be kind of tilted. For a long time, like when this was first observed in the early 2000s, this was really surprising that cells could fire in this like perfectly hexagonal way. And it kind of evoked this idea of a coordinate grid. If you think about like latitude and longitude, it was like, oh, this is the basis of latitude and longitude. This is how our brain kind of maps distance between places in the world, and then these cells input to place cells, and place cells kind of fire at the pinnacle of a set of these grids intersecting. Now, 2 decades later, there's been a lot of research. It's more complicated than that. Grid cells can also change their firing based on context. They can warp their firing based on rewards. So we'll probably get into this more in a moment because this is kind of my favorite feature of the whole system. And so if that's true, that means that they're not doing a perfect coordinate spatial grid. They're not just the brain's GPS, because a GPS doesn't warp based on the parts of the world that you care about the most. It's just the map, right? So what we do know that grid cells are important for is measuring distance. So even if there's this warping, they seem to be kind of measuring our steps. And this is the most evident in work from my postdoc lab at Stanford, the Geocoma lab. And what some of my colleagues had done was put mice running in virtual reality. This is also what I did in my experiment, but in their experiment, the mice were running in virtual reality. And when I say that for the listeners, they're not wearing little VR goggles, even though some groups have actually made that. Little mouse goggles do exist.
That would be cute.
But in this case, the mice are kind of in an IMAX theater situation. So they're running on a little treadmill. There's monitors around them that are displaying the VR. And my colleagues were recording in the entorhinal cortex. And if they turn the virtual reality off so that the mouse is running in pure darkness, the firing of grid cells will exactly map distance. So it'll be purely distance run. And then if you start to turn on parts of the virtual reality and kind of build up the scene bit by bit and add landmarks, the grid cells will start kind of anchoring to the different landmarks and developing this really structured firing pattern that is a combination of distance run and where you are in proximity to certain landmarks. So I think this system is really important for navigation. It inputs to the hippocampus. Hippocampus sends information back to it. And many computational labs are really fascinated with grid cells because you can, as I said, you can describe them with really elegant mathematical models.
So because I want to delve in a bit more into obviously that paper that was published in Nature Neuroscience, but with regards to grid cells, so first of all, you said they form 15% of the EC, correct?
Approximately, it depends on experiment, yeah.
What other cells are there? Are they also
Yeah.
important?
What's the other 85% of cells? Yes, they are also important. So the majority of them are kind of mysterious and what we call non-grid spatial cells. So they have some spatially organized firing, but it's not grid, it's not anything else. It's kind of like a blobby place cell.
Mm-hmm.
Another fraction of the cells are border cells. So they will fire at the borders of an environment, kind of like just straight along the wall.
Mm-hmm.
Or if you insert a new wall in the middle of an environment, they'll fire along that. So that's kind of cool because we need to, understand the borders of our environment. There are other cells called head direction cells, so they will fire when the animal's head is pointing a certain way. So let's say every time the animal's head points north, the cell will fire. If it points west, it won't fire and a different cell will fire. Then we've also got speed cells. So these cells will fire based on how fast the animal's running. And so this whole constellation of cells has kind of evoked this idea of like all the components of navigation. So you've got your head direction cells, which are your compass. You've got your speed cells, which are your speedometer. You've got your border cells. And then you've got your grid cells, which are thought to be your coordinate system. I think all of this is more complicated than that. And all of these, or most of these cells that have been looked at, are also very contextually dependent and not just a pure GPS. So even the head direction cells, there's a lab at NYU run by Andre Fenton that I think is very cool. And what they do is they have this task where an animal has to kind of switch its attention between the external environment and its internally defined reference frame, and the head direction cells will actually switch with it. So they'll kind of toggle between world coordinates and internal coordinates that are important to the animal. And so I think that's kind of nice evidence that even this head direction cell population is not just a compass, it's also contextually regulated by what the animal cares about.
So, side note, side tangent. So, I presume people have done single cell RNA-seq on these cortices
Mm.
to sort of look at all of these different cell types. And when just fascinated experimentally and trying to understand the roles, do they use just like, can they use like optogenetics to sort of understand the function of these cells and link it to a behavior phenotype? Like, how does one
Mm.
go and be like, yes, this cell is involved in this and this cell is involved in this, like experimenting? Because I'm a, cell biologist, biochemist. This isn't my thing. And I find it really interesting from a experimental design sort of view.
Yeah. So unfortunately, I don't believe that there is molecular differentiation between border cells and head direction cells, for instance. Grid cells are two different specific cell types. They can either be what are called stellate cells or pyramidal cells. So those are going to have some molecular markers. I believe relin is one, the protein that these cells specifically express, but it's not exclusive to them. And so I don't think you could, just optogenetically stimulate grid cells and no other cells. What can be done, so again, this was done by my postdoc PI, Lisa Giacomo, and her first grad student, Caitlin Mallory. The scale of grid cell firing, like how far apart their hexagon is actually regulated by a certain ion channel called HCN channels. And so what Caitlin did, which was super cool, is you can genetically manipulate this ion channel. And so she did a genetic manipulation of this ion channel and it changed the scale of how the grid cells were firing. So this is kind of a very elegant, specific manipulation where you're not necessarily turning off grid cells, but you are changing something about how they fire in space. And she saw that with this expanded grid cell scale, the animals were impaired at path integration. So path integration is this term for our ability to kind of count steps and know where we are in space relative to some starting point. So the kind of clearest example of this is, let's say you get up in the middle of the night in your own bedroom, you kind of know exactly how to walk out the door and go to the bathroom, right? Even
Hmm.
if the lights
Hmm.
aren't on, even if you can't see anything. So that's really good path integration. You've learned exactly how far and how you need to be oriented to go from your bed to the bathroom. If you wake up in the middle of the night in a hotel that you're staying in for the first time, you might bump into the desk or like not be as good at this. So that's kind of what the mice were like in the HCN manipulation, is that they were worse at that path integration. They were kind of like as if they had never, they were in the hotel and they hadn't seen the space before and they were worse at it. So I think that's one of the few examples that I can think of with kind of a cell type specific manipulation that uses some
Mm.
genetically encoded trait of these cells.
Yeah, I think I mean, easier said than done, obviously, but very, very neat to sort of do, let's say, a screen of impairing certain cell types and then trying to link it to a behavioral phenotype that you see and then sort of
Hmm.
put it all together and come and meet in the middle and see,
Yeah.
the sort of the function of each cell. But, probably we need better techniques to understand the molecular, let's say, profile of all of these cell types before sort of delving into that. So with regards to grid cells, do they communicate with place cells?
Yes, they do. They directly input onto place cells. Um, I believe the lab that discovered grid cells, the Mojo lab, I believe there's an experiment where they um, specifically inhibited the grid cells that were projecting to the hippocampus, and place cells were somewhat disrupted. Place cells will still be place cells even if you completely inhibit the entorhinal cortex. So this is kind of part of the mystery. Shut off the entorhinal cortex entirely, you will still have place cell firing, meaning that you will still have spatially localized firing patterns. But the place cells in the hippocampus will be less stable. They will change their firing patterns more between different environments, more than they should, even with the same environment. So you still disrupt it, but it's not, I guess, in the necessary and sufficient language that we use in neuroscience a lot. Grid cells are not necessary for place cell firing, but they are necessary for the stability of place cell firing, I guess.
Okay.
Yeah.
Okay, that makes sense. That makes sense. So do grid cells and place cells form this sort of cognitive map? It's a term that I came across,
Yes.
cognitive map, and I think I'm just blindly chucking it out there and just putting words together like a 5-year-old. But um, talk to me about cognitive maps and if grid cells and place cells will form that.
Yes, grid cells and place cells are thought to form this cognitive map. Now, what do we mean by cognitive map? Again, this depends on who you ask. Some people have interpreted this to mean like literally your brain's GPS, literally the map of space in your brain and nothing more. The way it was initially intended from Tolman and then John O'Keefe and Lynn Nadell kind of in the 1970s was this scaffold that you can tie different events to. So it's kind of your memory map. And this is how I think of it. It's the kind of like organization of events in your experience. And so one subset of that organization of events in your experience is space, right? Space also organizes events, but then you can tie emotions to it and people and rewards and, all sorts of other sensations can be tied to different events. And this cognitive map is kind of how your brain organizes your understanding of your past experience so that you can recall memories and create future plans and kind of understand how different things are related to each other.
So, is it fair to say that the hippocampus plays a role in forming cognitive maps?
Absolutely. Yeah.
So, how does this change during transitions from one experience to one experience as we age? And yeah, how does that sort of work? I'm sort of trying to. I don't know if that's clear enough as a question.
How does the cognitive map change as we age?
Yeah, and sort of
Yeah.
the relationship between all of these, like, how does the hippocampus deal with this? Because it's a lot. It's a lot of data from a
It is.
yeah.
It is. So I can comment on the aging question just in
Hmm.
terms of like what is experimentally known. I mean, always whenever we're translating some experimental finding from rodents to humans, we might not know the whole picture yet. So far, most of what we've seen in rodents, if we can test for it in humans, has been replicated in terms of hippocampal activity, but in aged humans especially, it's a little bit of a mystery. But there's kind of two cool findings from recent years in aged animals in terms of how hippocampal and entorhinal activity change, and then we can kind of speculate about what this means for the cognitive map through aging. So one finding is from, again, my postdoc lab, Lisa Giacomo's lab, and my lab mate, Charlotte, for her PhD, she studied aged mice. So mice live about 2 years, and so at around 24 months, that's like an 80-year-old person. She compared young mice, the equivalent of 20-year-old people, to middle-aged mice, the equivalent of 50-year-old people, to aged mice. And she recorded from their entorhinal cortex and looked at their grid cells. And it's a very hard experiment because you have to age these mice for 2 years before you can even record from them, and then you have to hope that they do your behavior and you can get good neurons and all of this. So it's a pretty heroic experiment. And what she found is that the aged grid cells had impaired contextual specificity. So what that means is that within a context, like within one environment that she gave the mice, they would change their firing pattern more rapidly. So they were less kind of locked to that context. They were just kind of spitting out spikes in a way that wouldn't be that informative to tell you about what context you were in. And then between contexts, they also didn't change their firing patterns enough. So it was, if you were trying to decode, which is often a thing we do in systems neuroscience, what context the animal was in from the firing patterns, you had a really hard time telling because they were just these mushy firing patterns that weren't discriminating between the contexts very well. Another paper from the last few years from Kei Igarashi's lab made Alzheimer's model mice. They knocked in amyloid precursor protein, which is a protein in Alzheimer's disease that forms these plaques that are a hallmark of the disease. So they knocked this into mice, and they saw that place cells had, similar to these aged grid cells, disrupted remapping. So remapping is a term that we use for when a cell changes its firing pattern from one context to another. So you asked about, if between Stanford and Cambridge, place cells would remap. They would have a different firing pattern for those two different contexts. Place cells in the hippocampus were impaired at this. So again, they weren't discriminating context as well as they should have, and they weren't encoding a single context as well as they should have. So if we kind of put these two findings together, we've got degraded contextual representations in entorhinal cortex, we've got degraded contextual representations in CA1. That would mean that you would have a hard time knowing where the events in your life map onto each other in your cognitive map. If you're not able to discriminate two different events, let's say you go to one party on one Saturday and then you go to another party on another Saturday and you're like, I can't remember who I saw at what party or where which party was because I'm not representing these two experiences clearly. that would map on to the symptoms that we see in dementia and Alzheimer's. And if you don't have stable activity, you also can't generate new memories. The activity would not be reliable and you wouldn't be able to recall it later in a way that makes sense because it's just firing all over the place. It's not, precisely encoding these experiences anymore.
So on this, because I know this is very systems neuroscience, but like, has anyone sort of dug in, let's say, mechanistically in the cellular level to look at, let's say, impairment of expression levels of certain channels, or if there's impaired autophagy or mitochondrial dysfunction, or any of these sort of organelles,
Mm.
protein, big keywords that may affect, let's say, the functionality of these place cells or these grid cells in these sort of contexts, or even as we age? Has anyone sort of looked at that?
Yes, this is less my area of expertise, so I'm not
Hmm.
going to be able to give a very detailed answer on this. One thing that we know degrades in Alzheimer's is interneurons. So,
Hmm.
interneurons in the hippocampus die. Now, interneurons are the inhibitory cells in the hippocampus. They're like the cells that release GABA and that kind of turn down excessive excitation. Sorry, my kitten is paying a visit. We know that these die, and we know that if you have too much excitation, that is a bad thing for the hippocampus. That prevents its ability to encode information precisely. In the most extreme examples, that leads to epilepsy. So that's one kind of particular cell type that's affected. And from sort of totally separate work, we know that the proteins involved in synaptic plasticity are also really important for all of these firing patterns. So, in one experiment that I was involved in, we conditionally, meaning only in the hippocampus and only at a specific time point, knocked out a protein that's important for vesicular exocytosis of NMDA receptors, and that changed how hippocampal neurons were firing in a very specific way. So, there's a lot more work about this, again, that I'm not an expert in, at what kind of trafficking proteins are important in plasticity and in stabilizing synapses. And if you manipulate these, you can impair all sorts of things about spatial coding.
And then a very, very out of the box question, but it just popped up because you mentioned parties. How does alcohol affect? Why is it, if I go to a night out and then the next day I'm not admitting to anything here, so I can't get convicted or anything. Um, if I don't remember anything from the last night, why is that? Is that affecting my play
That's a
cells or grid
great that's
cells,
a
do you
great
think?
question. Again, I'm sure that somebody has studied this. I know that there are groups that study alcohol and place cell activity. I don't know the results off the top of my head.
Hmm.
I know that in general, alcohol is thought to slow down neurotransmitter transmission between cells. So you can imagine if you've just disrupted the signaling between neurons in the hippocampus, you're not going to get a proper encoding of that experience because you're kind of scrambling the wires, if you will, between cells. So I think that's kind of the oversimplified explanation.
Yeah,
Again,
yeah.
not
I guess
not
it's
my
just,
area of
yeah,
expertise, but
scrambling circuits, altering the synaptic activity, and yeah,
yeah.
that makes sense.
Hmm.
So let's go back to your paper from your postdoc lab
Hmm.
about the. Would you say you identified two populations of cognitive maps, one which was more based
Mm,
on, let's say, spatial map, and the other one which was more related to reward and, well, reorganizing around reward? Because that
yeah.
was an observation that sort of progressed the field in how we sort of looked at cognitive maps, right? And how reward and value comes in and influences that.
Yeah, yeah, I think that is one really nice way of thinking about it. So, what I did was use virtual reality again to be able to dissociate the animal's experience in space from its experience relative to rewards. So, the example that I always like to give to kind of explain this is, let's say you're walking down the street in your neighborhood. I used to live in downtown Palo Alto. So, let's say I was walking down Lytton Ave, and I knew, I know where I am in space, I know where I am in Palo Alto, and I also know that my favorite coffee shop is on a particular street corner. So as I'm approaching this coffee shop, I know, not only am I 50 feet away from the intersection of Lytton and Cowper Ave, but I'm also 50 feet away from this coffee shop. Now, let's say that this coffee shop is closed on one particular day, and so I have to update my current knowledge of where reward is and continue down the street to another cafe. And I find, I go into this new cafe and I find, oh, I really like this one too. This is also great. I basically now have two maps in my brain. I have a map of where I am on the street, my spatial map, and I have a map of where I am relative to each of those cafes. And that map is representing both of those cafes similarly because they're rewarding experiences. So the way I simulated this for the mice is that they had to run down a virtual hallway and there was a hidden reward location. So hidden meaning that it didn't have an explicit visual cue telling them where the reward was. They just had to learn from experience where it was and use all the other landmarks in the environment to kind of extrapolate where it is. And they could remember where it was. And then I would move that reward to a new location, again with no explicit cue, just now it's in a new place and they have to update their knowledge of where it is. And so what we saw was that in the hippocampus, and we were using a technique called two-photon imaging to image a very large population of neurons at once. A subset of the neurons would maintain their firing in space relative to the virtual context when we moved the reward, and a subset of them would remap, which is this term I mentioned earlier about updating their firing patterns, and they would remap to the same place relative to the reward. So if that cell fired 100 centimeters before the reward when the reward was in its first location and we moved it, it would still fire 100 centimeters before the new reward. And so across the entire environment, you would get this complete map of where the animal was relative to the reward in a subpopulation of neurons. And so we kind of interpreted this as one way that the brain could kind of hold in memory, let's say in that cafe example, where you are relative to the city block and the street and where you are relative to your favorite cafe. And you can kind of use those pieces of information interchangeably depending on what goal you have. If your goal is to find the cafe, you could rely, and you've gone there many times and you know exactly how far away from it you are, you could exclusively use that reward relative population. But let's say something changes in the environment, like there's a road closure and you need to navigate around it and still kind of know where in space this cafe is, you would still need your spatial map to be able to do that.
Okay. So to summarize, you have one population which is sort of stable. It provides the groundwork, let's say, like a
Mhm.
nice sort of
Mhm.
map. And then you have this other population which is reward related.
Yeah.
And that doesn't just fire strongly close to reward, but relative to a reward. Or
Right.
is that the same interpretation? Did I just say the same thing twice?
No, they're slightly different because you could have the stable spatial map also fire more
Hmm.
when the cells are near reward, but you don't. You only have the cells that encode where you are relative to reward do that. So only that population of cells that has this reward relative map also increases its activity near the reward. And the way that it increases activity is that over learning, more and more neurons are recruited from the whole hippocampal population to fire close to reward in this reward relative sense. So the way I think about that is kind of like as the animal learns more and more how to compute where reward is in this reward relative reference frame, that memory is being amplified. There are more and more cells contributing to it. So if somebody was like listening to hippocampal activity and saying, "Hey, where's the reward?" more and more neurons would be shouting back, "Here it is," as the animal learns.
Okay.
And so, yeah.
Sorry. No, please. I guess one thing that just popped up to me is so one thing I did, I struggled at school because I hated it. And, I found it difficult as well when I was doing my undergraduate at university. But one of the sort of things, let's say the hacks I realized that I could do and I implemented was gamifying tasks to make
Mm,
them more fun.
mm-hmm,
And that helped me learn a lot of information in one go. And
mm-hmm.
I could, I, I switched from not being able to recite textbooks to reciting textbooks. And it wasn't
Wow.
just through hard work, but gamifying tasks. So
Interesting.
yeah, what do you think could be the relationship between, let's say, boring stuff, but also enhancing your memory of the boring stuff by trying to integrate reward and value into it?
Yeah, I mean, it sounds like you kind of switched your manner of learning information from a kind of purely factual, maybe semantic type of learning to something reward based, right? Like something with a goal that gave you a reward at the end of it. And so, I mean, in addition to, increasing the recruitment of hippocampal neurons, which is kind of what I showed, all over your brain, this probably did all kinds of things. It might have released more dopamine because you were more motivated. It kind of, creates this ramp leading up to that outcome that can help associate all the information along that path more strongly with the outcome and then help encode that better in your brain. You probably increased your acetylcholine, which kind of governs your attention to a specific task. I think reward is a really powerful mechanism throughout the brain for learning. And, many decades of research have backed this up. And I think a lot of the field would agree with me. And kind of for whatever reason, only recently has it been gaining a lot of attention in the hippocampus because there was this idea for a long time that the hippocampus is just doing this like spatial coordinate system. But now it's becoming clear that it really changes its activity based on what the animal cares about. So when you made yourself care about what you were doing by gamifying it, you probably, improved the robustness of how your hippocampus was responding to the task and improved that memory encoding by making it interesting for yourself.
So would you say that the hippocampus obviously is always currently updating its internal model, but
Hmm.
is it doing that due to actions before that or concurrently or after? Like when is it, if from a sequence of events, when is it always, or is it continuously updating? Um,
Mm,
or is it dependent on rewards and the certain context and, because,
interesting.
yeah.
This is a good question. Hmm. I think the hippocampus is always making predictions. And so I think that means that it could update before an outcome if needed. And kind of the way I interpret my findings is that that population of neurons that kind of amplifies its activity around reward is making an internal estimate of where the reward is. And so that's why we saw after we switched a reward location, that internal estimate would update to the new reward location even before the animal's behavior fully solidified at that new reward location. So by behavior, just to be really concrete, the animals do this anticipatory licking because the reward is delivered from a little straw in front of their face. And so they'll kind of lick at that straw even before they get the reward. And that's just a classical sign of understanding that the reward is coming based on predictive features like where they are in space. And so that licking and their kind of compensatory changes in running speed are congruent with that because mice can't run fast and lick at the same time. Neither can we, right? You can't like eat an ice cream cone while you're running a marathon. So they slow down when they lick more. Those behaviors would kind of gradually shift to the new location, even though
Mm.
the neural activity would shift quickly. So the neural activity would update, encoding the new location in space. And I think that reflects an update in their internal estimate even before they had fully received enough outcomes to tell them that, yes, this is where I'm now going to be getting reward. So I think your question is a really good one. And I don't know if we have a full answer, but there's a lot of, not just my research, but a lot of others that have kind of suggested that, the goal of the brain is to make predictions about our experience because that's what's evolutionarily advantageous. Like we can't adapt our behavior just based on whether the lion eats us because then it's too late, right? We have to kind of be able to update our behavior based on predictions of what the future will be. And this kind of ties back into what I said about the hippocampus and imagination and being able to predict the future. I think it's able to update its activity even before there's an outcome. And then you get an outcome and that kind of updates the prediction, and you kind of work backwards from there.
This is going to be a weird analogy or similarity, but I'm thinking of like a bacterial growth curve, where you have sort of that exponential growth phase and then a saturation point almost. Would you say potentially that you would have that sort of a brief increase in neuroactivity, and that would be sort of an early predictor with the mice licking whatever, and then as they then keep on going towards that reward, then you sort of have more increase in neuroactivity, and then saturation, it reaches this sort of threshold? Is that too simplistic or completely wrong or too much of a wild stretch?
It's not a wild stretch. That's not exactly what happens in the hippocampus
Okay.
because actually the majority of the reward relative firing was slightly after the reward location. And that's why I think it's a little bit more consistent with an internal estimate because slightly after the reward location was the highest probability of getting reward.
Hmm. Hmm.
So I think it's a little bit more about that estimate of probability, whereas what you're describing, this kind of ramp to threshold, is maybe more what you would see in dopaminergic release leading up to this location, this kind of ramp that reaches its peak at the outcome. And that's been seen, I guess, both a little bit in the hippocampus and in areas that receive a lot of dopamine, like the ventral striatum, as you see this kind of ramp. This ramping to threshold idea is also something that's seen in monkeys and certain cortices that are important for decision making. So as like evidence is accumulating, you'll get this ramp in firing rate that reaches this threshold when a monkey makes a decision. So I think there are other brain networks that do operate like
Mm,
that.
mm.
Yeah.
Yeah, because I'm not sure if you can answer this, but obviously earlier on you talked about the hippocampus and its links to the VTA with regards to dopaminergic signaling. But because our estimates of rewards or our value that we put on certain rewards can change throughout our life, right? And that can change in people with drug addictions to people who now just mindlessly scrolling on Instagram, who's always getting this sort of continuous dopamine hits. So how can dopamine's role in, let's say, altering the value of certain rewards then have an effect on your map, on that specific reward related map? Because I don't know, maybe 5 years ago, I loved going to a coffee shop in Cambridge. That was my way to work. I loved it. And then maybe a couple months later, I got bored of it and I didn't like it because I like tea.
Yeah.
My rewards change. And so it's a very complicated question. I don't think there's an answer, but
Right.
like, how does all of this sort of come together, do you think, when you're looking at not just the HPC EC as one system, but the interconnectedness and how all of these systems can influence rewards and memory?
Yeah. Yeah. Well, as I said, I mean, even dopamine doesn't have a strict one-to-one relationship with reward itself.
Hmm.
So, and to make that even more complicated, there's individual variability. So there's kind of this classic set of experiments where some rats are what we call sign trackers, meaning that they'll have more dopamine release for the cue that they think predicts a reward. So this might be like the people scrolling on Instagram, maybe they're sign trackers. They just have this like dopamine hit every time they like open Instagram and like flip to another reel, versus goal trackers. So another set of rats will have more dopamine release when they go to the location of the where the reward is delivered, and they don't pay attention as much, or they don't care as much about these cues that predict the reward. So people are probably like this too, right? We probably have all over the spectrum of how much we value the stimuli that we think are going to lead to reward, and we just need that like constant scrolling, versus maybe that doesn't do that much for us and we need the outcome, and we can change what outcomes we like, and those can motivate us in different ways, whether we like coffee or tea. So I don't know if that's a one-to-one relationship with sign tracking and goal tracking,
Hmm.
but I think it can be really individually dependent. And, there are other systems in the brain that help govern what kinds of things bring us reward, serotonin, norepinephrine, all sorts of things. So I think it's really complicated. And you asked about aging. We know that dopamine is very susceptible to degradation in Parkinson's disease. So this is kind of something that kicked off our conversation with each other, was dopamine's role in Parkinson's and dance. So I just want to make sure to mention this because I think it's super cool. So you kind of found me because I participated in this dance performance at Stanford on neuroscience. It was a dance show about neuroscience, which was honestly like one of the highlights of my career, because as a former serious ballet dancer, getting to kind of match my passions between dance and neuroscience was very exciting. And there was even a moment where the choreographer had asked for audio visuals to go with the performance, and I had sent her some of my 2-photon calcium imaging of hippocampus, like a video of neurons firing, and it was playing on screen behind me as I came out on stage, which I wasn't expecting. So I got a very big positive reward prediction error from that. I wasn't expecting it. It was very rewarding, and I was like, wow, this is great. To kind of close the loop here, is an effective therapy for people with Parkinson's disease. It's not totally clear why this is. I did a little bit of extra reading. There's just not enough research to know why this is effective. But one hypothesis is that dopamine neurons die in Parkinson's disease. They specifically die in a brain region called the SNC, or the substantia nigra pars compacta, which is right next to the ventral tegmental area. And the SNC sends a lot of dopamine to the striatum that is important for governing movement, for initiating movements, for completing sequences of movements. And so these are the neurons that die in Parkinson's. And so people with Parkinson's will have this kind of short shuffled gait where they have trouble initiating, walking, and moving, and they are very stiff and rigid, and they can't move very well. And one hypothesis for why dance overcomes this is because it taps into auditory and rhythmic cues in the brain. So it engages the auditory system to almost bypass this dopamine needing initiation of movement to kind of directly trigger the motor circuits that can trigger movement with music. And so I remember many years ago seeing this really beautiful video of a Parkinson's patient who couldn't walk down the street from his house to, the nearing cafe by himself. But if you gave him music, you put on headphones and you told him, "Can you dance down the street?" he could get there. So he could like use the music and it also, of course, is going to evoke joy and confidence and kind of self actualization and ability to move again and all these things. Parkinson's patients go to dance classes. There's a social component, which is really rewarding. And so maybe that's kind of activating these other reward systems in the brain that might still be intact, maybe serotonergic or something else, along with using the music to kind of cue movement and overcome this, inhibition of movement.
So building on that, because I
Yeah.
was going to bring up music and dance, obviously, as a musician myself. Do you think it provides almost this alternative compensatory pathway to motor, let's say, action, aside from the main circuitries involved in motor movement? Is that sort of
I think
so
that's
almost
the
like a
idea.
hack, right?
Yeah.
It's a hack
Yeah.
almost. And then my second point to that would be, could dance or music, would it just have a positive effect on, let's say, ex dancers or on people who haven't danced before? And the reason I sort of bring this up is because, you know, earlier on in the podcast you mentioned episodic memory,
Mm,
and if I'm correct in saying, I don't know, like some patients, they can remember what happened to them when they were 5 years old, but not sort of 5 minutes ago, right?
right.
And so, if, as a musician myself, I remember when I was 10 years old, just it being drilled into me, certain movements, could, one sort of hypothesize that in these patients who are ex dancers, they can sort of use that memory of movement 50, 60 years ago, and again use that as a hack to then bypass and help motor movement now, if that makes sense.
Yeah, this is really interesting. So, to answer the first part of the question, even in people who have never danced before, dance therapy is effective for
Mm.
Parkinson's. But if whether it's more effective in people who used to be dancers, I don't know the answer to that, but I would imagine yes. I would imagine that music and that, triggering that long-term memory, and it's often kind of a muscle memory, but it's maybe a muscle memory that's laced with positive emotion, could help overcome that, movement rigidity even more. And I think, in dementia, sometimes dementia and Parkinson's are comorbid with each other and occur together. There's also evidence that, former musicians or former dancers, if you put on a piece of music that they used to know, they will kind of come to life. They will, remember the lyrics to the song, or they will, start moving their body in a way that, repeats the choreography that they learned long ago. I'm forgetting off the top of my head who it is, but there's a beautiful video of an ex principal dancer from the New York City Ballet who now has Alzheimer's, and she's, normally very like subdued and kind of, depressive affect and not talking. But if you put on Swan Lake, she starts moving her arms to the exact choreography from, the Black Swan dance that she used to perform many decades ago. And so there's something about that music and dance. I don't know where that is stored in the brain, but that seems to be preserved longer than that kind of ability to form new memories and remember what we did 5 minutes ago.
But I mean, like, if you were to put that, woman, I don't know the techniques, but under fMRI machine, what
Yeah.
what would be activated?
I have no idea. Who knows? Maybe it would activate the hippocampus more than her hippocampus would normally be active. Maybe it would activate, the reward centers of the brain, like the ventral tegmental area, nucleus accumbens. It would probably activate her kind of auditory cortex, language cortex. I don't know. I think that'd be a very cool study. I'm just making guesses.
Yeah, because I think what I'm trying to get across and trying to think out loud is that, you mentioned again earlier that episodic memory, it's different to obviously semantic memory, I think, because that's on facts, where episodic memory takes into account the context of environment and emotion and, all of these sort of responses that make us human in a way and different to, let's say, AI for now, anyway.
Mm, mm,
Um, so, if you coming back to your Nature Neuroscience paper, if we were to not only integrate, let's say, music or dance, but also like change the location using VR,
mm,
for instance,
mhm,
and we add, I don't know, certain smells or whatever to kind of recapitulate that sort of environment in a way.
mhm,
Could
mhm.
that be, I don't want to say beneficial because that's jumping the gun, but could it help sort of with certain compensatory circuitries in patients with dementia or Parkinson's disease? Again, this is
Yes.
completely hypothetical.
Sure, sure. No, this is a great idea. And we know that cues that are associated with the initial experience, like the context and the music and the smells, trigger memory recall better than if you're just trying to remember it in isolation. This is in like technical language, this is called contextual reinstatement, that you provide the context and you trigger the memory. That's a little bit of a tangent. This can be maladaptive. I'll come back to your question in a minute.
Hmm.
In individuals who are recovering from substance abuse disorder, actual reinstatement can be a problem because they have this really strong association between some spatial context and some set of cues and seeking the drug.
Hmm.
And so
Hmm.
let's say someone has their first drug experience at a concert, they might be in recovery, they might be, better in a lot of ways, but if you take them to a concert again, they are most likely to relapse because of that contextual reinstatement. But it can be also, used beneficially in this, in thinking about dementia and degradation of memories. I'm at University of Colorado Boulder now, and we recently had a guest speaker. Her name is Morgan Berence, and she is a professor at University of Toronto. And she has come up with this smartphone app called Hippocamera, which I think is so cool. So she works with humans, but she has taken inspiration from the rodent literature and this phenomenon that I mentioned called replay, where the hippocampus can replay memories in this time compressed format. And there's some evidence in rodents that if in rodents that are a model of Alzheimer's, they have degraded replay, and maybe if you could enhance replay, you could kind of rescue memory deficits in dementia and Alzheimer's patients. And so she took inspiration from this. And so what this smartphone app does is she asks the participants to record one event per day that they think is interesting in their daily life. And so they'll take a video of it and then they'll kind of narrate, during the video after they kind of rewatch it, what the event was to them. And Hippocamera will take these little snippets of the event, kind of key video frames, key cues and moments, and compress it into like an 8-second little replay reel. And the goal is to have people watch these little replay reels and then test them later and say, do the participants who had this replay visual cue, these kind of contextual reinstatement of these little pieces of the memory to help them remember it, are they actually able to recall other details of that experience better than people that did not have this cue? And the way they test this, which is it's a very well-designed study, they score the details that they can recall that are not in the video, right? It would be cheating if you just said, how well do you remember the details in the video because you just saw the video. But if, there was this example of they record, it's a grandmother recording her kids playing in a lake. And you score her based on what other details she can recall from that experience. So if, in the video, it was just her son in his bathing suit, her grandson, like going into the lake and taking a bucket and trying to scoop up water and look for minnows. If she says things about the bucket and the minnows in the recall, that's not informative. But if she says, that was the vacation where we stayed at our family's lake house and Jack had just found a water snake and he was really excited about the water snake. And so he got his bucket because he wanted to see what other creatures were in the water. And then after that, we went inside and we had peanut butter and jelly for lunch. So all those other details tell you that she remembers that episode really vividly now because she's had that replay experience compared to maybe someone who didn't have that kind of replay experience or compared to other memories that she didn't capture with this app. So I bring this up because I think this is a really cool application of all this rodent basic neuroscience to something that is going to try to leverage that activity in the brain to help people. And so kind of getting back to your question of could you, provide someone all of these cues from their original experience to help improve their memory? Like this is maybe exactly that type of thing. So I think that's a, kind of exciting direction for the field to come up with these kind of therapeutic, techniques that are compatible with humans to try to boost their memory. And I think the hope would be that by boosting hippocampal function by providing these cues, you would also be able to boost, it would spill over into their ability to encode new memories too, which is the main thing that's inhibited in dementia.
I think the answer is no, but do you think in those situations, in let's say in the disease context, do you think there's still an element of neuroplasticity that could be leveraged in a way, or is it completely nada?
I don't know. I hope that there is.
Mm,
I think it really depends on what state the disease is in, how far it's progressed, and, the specific pathology, whether it's the death of interneurons or whether it's something more at the level of, the individual excitatory cells and their ability to have synaptic plasticity. I mean, if the interneurons are dead, you can't bring them back to life,
hmm
right?
hmm
Unless you infused stem cells into the brain that were going to differentiate into interneurons or something, which, is a
Yeah,
therapeutic that many groups are working on. So
yeah, yeah.
with something like that, maybe you could, if you rescue that function, maybe there is, if the GABAergic receptors on the pyramidal cells haven't degraded yet or are still there, then there's still that mechanism that could allow the interneurons to do their role once
hmm
they've been replaced. Yeah.
hmm Yeah, because the cell biologist in me who nearly actually started a company on this with a very good friend of mine. We were looking at
Cool.
trying to see, okay, how can we help this through stem cell transplantation, or can we harness the mitochondria pathways and mitochondrial
Mm-hmm.
dysfunction, like from a cell biology perspective, what can we do? But then
Yeah.
there's the other perspective, which is more on a clinical neuroscience sort of lens. And this is where my love for music and neuroscience comes in, where can we use deep brain stimulation? Can we try and identify these sort of alternate compensatory circuitries and target them and use that in combination with, let's say, more behavioral therapies to have a better therapeutic outcome in conjunction with pharmaceuticals? So that's sort of the rationale as to why I'm sort of interested in how we could marry all of these perspectives together
Mm-hmm.
and to create, let's say, cohesive, not research program, but cohesive therapy for patients who are suffering from neurodegeneration. But obviously, I think it's very ambitious and
It's ambitious,
requires.
but I think it's the right idea. I think like with really complex diseases like this, one type of approach is not necessarily going to be sufficient. So if you can combine approaches, if you can combine some pharmaceutical or stem cell therapy or something with a behavioral therapy to recruit that neural circuit that you're treating with the pharmaceutical in the right way, in the context of the brain networks that are associated with the behavior, it might work better than if you just give that pharmaceutical alone. It's like one idea that's coming to mind. I'm not a Parkinson's disease expert, so I don't know what the state of the field is on this research, but I remember learning in grad school that there was some thinking that mitochondrial dysfunction in the dopamine neurons was leading to their death. So if you could rescue that in some way, if you could identify what this molecular mechanism was of the mitochondrial dysfunction, provide some pharmaceutical that could help slow that dysfunction or interrupt whatever the bad process was, and then also give the patients dance therapy, would they actually recover their dopaminergic motor function even better because you're activating the downstream motor circuits by bypassing it with the music? It's a social experience, it's a rewarding experience, and so you're engaging all these reward systems in the brain. With that drug on board, would that work even better than the dance therapy alone or the drug alone?
No, that's well, you just summed up my dream research position.
Yeah.
But I mean, do you think, because I'm not a systems neuroscience expert, do you think it is possible to try and identify these alternate circuitries in, let's say, music therapy or in dance therapy, and then supplement that with neurostimulation? You're mute.
Sorry.
That's right.
Yeah, I think it's possible. I think the field could eventually get there. I think either with kind of new techniques in neural recording in humans,
hmm
we could learn about what pathways are active there. I'm thinking about a researcher who I really like, Nantya Sutana, who's now, I believe, at UNC. But she works with epilepsy patients. She's kind of the modern version of Brenda Milner, I guess, like looking at what neural activity is going on in the brain in awake behaving humans, which it sounds like absurd to say awake behaving, but like we talk about awake behaving mice, right? But she works with patients who have neural implants in areas like the hippocampus, And those implants are usually in there because they are epilepsy patients. And so they're, the medical professionals are trying to track what parts of the brain are having seizures in their daily life. But while those implants are in there, she can record from these patients doing other things. And so she records from humans who are walking around a room with virtual reality goggles on, which is super cool. So she can look at, how neural activity is actually functioning in the brain in people in their, actual movements and their actual life outside of the hospital setting. So maybe with a technique like that, not necessarily her specifically, but other researchers who do similar things could look at, what neural circuits are active in the brain in a Parkinson's patient when they're dancing, for instance.
That'd be such a cool experiment.
Very cool.
Oh, that'd be so cool. God, that's got me excited.
Because, there are Parkinson's patients who have deep brain stimulation implants because that's another therapy is to
Hmm.
to stimulate motor centers. So if, in those same devices, you could have recording electrodes
Yeah,
and
yeah.
have
Yeah,
the patients go to dance class and then see how that circuitry is responding during dance. That'd be pretty cool.
that is a grant idea right there.
Seriously.
No, this has been fun because I'm aware of the time, and it's been a pleasure to have you on, and
Thanks for having me. It's been really
hopefully we can
great.
have another conversation because this has been really fun, and like I really love this topic. Like I'm a cell biologist at heart, but I think I'm a disguised systems guy as well
Yeah, I
underneath.
think so. I think so. You really picked up on the nuances in the hippocampal literature in a way that, I think demonstrates your innate systems way of thinking.
Yeah, yeah.
So, thanks for the great questions.
No, thank you for coming on, and yeah, and great work as well. We'll put the papers in the link as well
Thanks.
and share it because it was very difficult to read. I'm not going to lie to you, but a pleasure to read.
The secret of those papers is they have a strict word limit, so a lot of content gets compressed into a very short format in order to say everything that you want to say.
But I think that's sort of the nice thing about podcasts is that you have this sort of not freedom, but you can sort of
Space to unpack. Yeah.
yeah, in
Yeah.
a way, especially with someone as questionable as me asking questions all the time. But yeah, no, thank you for coming on.
My pleasure. Thanks for
Yeah,
inviting me.
thank you. And this, yeah.